of all this information about epilepsy, tying the two together. So there are two major classes of changes. So you can have copy number variations, or basically in your DNA you can have some parts that are missing or some parts that are duplicated. There's also single nucleotide variants where you might have just a base portion altered. I'm just going to kind of show you words to kind of, or names to have you familiar with the words. I guess so Sanger sequencing is a form of genetic testing. It's highly accurate but very time consuming. Next generation sequencing, and these are the studies that we're really trying to do more of and try to give, and these are the studies that are giving us more information. Basically what it does is it takes large numbers of DNA segments and rapidly sequences them. It's very time consuming. So that's why if any of you have had genetic testing, like whole exome sequencing, which is kind of right now it tests everything we know about the genes and diseases and disease-causing genes. This takes about 4 months to come back, and it's extremely expensive. So this is upwards of $10,000. Some insurance companies cover it, which is wonderful, because it can really, especially in situations where it can help change therapy. Most insurance companies don't.
Which are the ones that do?
I actually don't know that information off the top of my head, yeah. It's one of those things where you could probably call your insurance company and ask. It doesn't cover non-coding areas, so this is a big, so we're still kind of at the tip of the iceberg for genetics, because what we can test for is all of the genes that are expressed. We have a lot of other genetic information that doesn't express anything, and that could be important as well. We don't know that yet. And also this type of, so even the whole exome can also miss certain types of genetic changes. So this is the International League Against Epilepsy. This is kind of an international organization, and this is how they recommend to go about genetic testing, and this is for adults and children, basically for drug-resistant epilepsy specifically. So we've tried 2 medications and things are still continuing. Obviously biochemical testing, so this means testing for metabolic conditions like with a spinal tap or blood tests. And then they actually recommend sequencing single genes in specific situations. So if you have a child that really looks like she may have Rett syndrome, you would specifically test for Rett syndrome. For tuberous sclerosis, it's quite easy because the genetic test is almost a confirmatory test, because you just test for that specific gene. There's something called a chromosomal microarray that we often do for children with learning problems or children with epilepsy that we can't treat with medication. And this is just another test looking at genes. Then there are gene panels, which actually look at specific genes associated with epilepsies. And then there's whole exome sequencing, which is a big test that looks at everything. And then they even consider, and I do this frequently with our genetic department. If it's been a couple years since someone's had whole exome sequencing, I'll check with our genetics team to see if anything's new. And so with single gene sequencing, so just testing for a single gene that we think could cause Dravet syndrome or Rett syndrome or TSC, the pros is that it's very efficient, it's quicker, it's less expensive, and it's easy to cover with insurance. But the cons are, it can actually be more expensive than panels. Multiple genes can cause the same type of epilepsy, so we're finding ourselves getting the panels more. And so chromosomal microarray may not detect certain types of genetic changes. So this is kind of a test that's looking to make sure, if you think of the encyclopedia example again, that all the chapters are in the right order. So it's not as fine as a whole exome sequencing that might see that all the words are in the same order, but this is all the chapters are in the same order, so this may not detect everything. Actually, it's a fairly high yield, about 23.5% might give us a diagnosis or a hint of a diagnosis, and especially so when multiple findings are present. So maybe if children have not only epilepsy, but let's say they have ears that are a little bit lower, they have heads that are a little bit smaller. Sometimes that'll help lead to a diagnosis as well. And a karyotype is kind of is making sure that all the volumes are there. Can detect changes not detectable in the chromosomal microarray. So we may get these two together, and these are 2 of our first-line tests that we often do in children. And this is an example of a karyotype. So what you actually do is it's a blood test, and the geneticists actually can look at the chromosomes on a microscope, and they kind of basically, by how they look like, they make sure that you've got 2 copies of each chromosomes and all the genes are in the right order and there's no pieces that are missing. Sometimes we'll find that some chromosomes can form rings, some chromosomes can have deletions, sometimes people might have 3, so trisomy 21 for Down syndrome. You might actually have 3 copies of chromosome 21. You can't really see which one's 21, but you might have 3 copies. So it's a helpful test to lead to a diagnosis. And gene panels, there's multiple companies out there. I don't have any that I particularly recommend, but we often switch back and forth, and our institution has its own genetic panels. But the quality really just, I don't think we have a sense within our institution that there's one company that's better than another. So some families have come to me for Gene DX, for example, or Athena, for example, and we say, we just look at what the panels have. We think about what the kids have experienced and what their clinical symptoms have been, and then we kind of pick which we think, could be the highest yield in the panels. So some are kind of 20 genes, some are over 400 genes, and some companies report that their genes have a 22% sensitivity, and then whole exome. We actually, in our institution, we found that it's been about a 30% yield, so about a third of the time we actually reach a diagnosis. So this can be really helpful. So there are limits, and this is the one thing I do want to mention with this study, the ethical concerns. So oftentimes what happens is that we're testing for epilepsy. We get a big gene panel, and then we find out that our child has a heart condition. You're like, oh, did I want to know that? And so now we know that we have to get echoes every year. We have to, we diagnose a condition that we weren't looking for. And so that's something that we always try to carefully talk about before getting this study. And unwanted data, for example. And you usually do have the option, if you don't want to hear about particular conditions, you can say, I only want to know about the things that are related to the symptoms.
You talked about microcephaly as being part of that earlier. Have you tested any of these children that are coming up with microcephaly from the Zika virus to know what kind of gene changes that virus may cause in children so that you can be prophylactically treating them for possible?
That's a really good question. We don't know very much about Zika virus now, other than the association with microcephaly, but we don't understand how it happens quite yet. And if it's a change that happens during pregnancy, it's not something that we can often change after birth, if that makes sense.
Well, I know you can't change it, but
you're saying during pregnancy?
Yeah, during or just after birth, that if they could be tested, genetically tested to see. I'd be curious to know, since you're talking about the microcephaly as part of the syndrome or whatever, that you can test them to see if that they're going to develop, or do you know if they develop epilepsy?
I don't think we know yet. We don't know at all. I mean, we think that for Zika virus, it's something, it's almost seeming similar to some of the other viruses that can cause infections in utero, like CMV, rubella, like those types of things. And what we found is that those actually don't result in genetic changes. They just result in, for whatever reason, they get in the way of the brain forming correctly.
Oh, okay. Well, that's
a really nice question.
Yeah.
Oh, yes.
I just wanted to add, my daughter's 8, and we started having genetic testing from the time she was 2 months old.
Oh, wow.
And everything kept coming back normal. I mean, they would just assume she had this and had that, and our geneticist kept saying, we're coming along every year, we have new testing, new things we test for, and this and that. When she was 5, she had the whole exome sequencing for something he thought she had. She did not have that, but we finally found out the rare syndrome she had. So I just wanted to say, even with some of the testing, when you think, because we're all human, and the doctors are like, she's got to have, Angelman's. They just assumed she did, but she didn't. But just by chance, we were able to diagnose her finally. And it's such a rare syndrome that they are now finding adults in their 50s.
Which syndrome is it?
It's Coolin-DeVries syndrome, with a K. And it was just, the syndrome itself was just discovered in 2006 as a syndrome. And so they're still learning, but it's interesting how many adults have lived over half their life and have it because the testing wasn't available. So I'm such a